Hello, grade 12 students. I hope that you're doing well. In this video, I'll be talking about oogenesis, which is the female gametogenesis. Gametogenesis is the biological process by which a diploid cell undergoes mitosis, growth, meiosis, and then differentiation to form haploid gametes. Spermatogenesis is the process by which sperm cells are produced from a spermatogonium. I will do a quick revision of spermatogenesis using the diagram explaining oogenesis to show you how similar they are. During spermatogenesis, a spermatogonium found in the testicles of males undergoes mitosis, then growth to make spermatocyte 1, primary spermatocyte. Then this primary spermatocyte undergoes meiosis 1 to make secondary spermatocyte, 2 secondary spermatocytes. Each secondary spermatocyte will undergo meiosis 2 to form 2 spermatid. So as a result, we will have 4 spermatids. These spermatids will undergo differentiation, so they will differentiate into 4 sperm cells, okay? This process is very similar to what happens during oogenesis, as during this process, which is the process of formation of ova in the ovaries of females, an oogonium, which is the mother cell, it is diploid. This oogonium will undergo several processes to give at the end an ovum or a mature egg cell. An oogonium will undergo mitosis, so it will multiply. So there is a multiplication phase. This oogonium will grow. During the growth phase, DNA is replicated. Then we'll get an oocyte 1. The oocyte 1 will do meiosis 1 to produce an oocyte 2. The oocyte 2 will do meiosis 2 to make an ootid, and then the ootid will mature and differentiate into an ovum. However, there is a main difference between spermatogenesis and oogenesis is that spermatogenesis is a continuous process, while oogenesis is divided into 3 big parts. Okay, a primary oocyte is produced before birth. After birth until the age of puberty, nothing almost happens in the female's body. At puberty, meiosis 1 and 2 resume. So we will get an oocyte 2 blocked at metaphase 2, and after fertilization, an ovum is obtained. So there are 3 big phases before birth, at puberty, and after fertilization, and we will tackle these phases in details in this video. So at the beginning, let me remind you that oogenesis is the process by which an oogonium, which is the diploid mother cell, will undergo several processes to produce the haploid ovum. Okay, this is not a continuous process as it starts from the fetal life. So while the female is in her mother's belly until menopause, from puberty till menopause. At menopause, which is almost at 45, 50 years, at menopause, oogenesis stops. The first phase of oogenesis is the multiplication phase. This phase increases the number of oogonia present in the ovaries. So the number of oogonia increases by mitosis, and several oogonia are produced. These oogonia have 2 N chromosomes, but with one chromatid. During the growth phase, the oogonia will give rise to larger cells, and these cells have 2N chromosomes with 2 chromatids. So during the growth phase, the DNA is replicated. This oocyte 1 will undergo meiosis 1. So till now we have oogonium, one oogonium. This oogonium, by undergoing mitosis and growth, will give oocyte 1. Oocyte 1 then will do meiosis 1. However, this meiosis 1 is blocked at prophase 1. This happens before birth. So all females at birth have their oocyte 1 blocked at prophase 1. We call this the fetal phase before birth. So all of these events happen in fetal life of the female. Start there and there. At birth, all girls have oocyte 1 blocked at prophase 1. Okay? Till when? Till a couple of days before puberty. So at the age of puberty. At puberty, oocyte 1 will continue meiosis. Meiosis 1 continues, so we will get oocyte 2. Meiosis 2 continues, but stops at metaphase 2. So at puberty and before each ovulation, what do I mean by ovulation? Ovulation is the release of an oocyte 2 into the fallopian tube. So ovulation is when the ovary releases an oocyte 2 in the fallopian tube. At puberty, before each ovulation, oocyte 1 oocyte 1 continues meiosis 1, finishes it, undergoes meiosis 2, but stops at metaphase 2 of meiosis 2. So each month we have an oocyte 2, which has N chromosomes having 2 chromatids. Since these cells, they finished meiosis 1 only, and they moved toward meiosis 2, but they were stopped at meiosis, at metaphase 2. Now, ovulation is the release of oocyte 2 in the fallopian tube. Now, there are 2 ways. The first one, if there was no fertilization during or within 24 to 48 hours, so the oocyte 2 will wait in the third part of the fallopian tube for 24 to 48 hours. If fertilization does not occur, so oocyte 2 will degenerate, we will lose it, and will move down, we will lose it into the uterus as the form of blood. What happens if fertilization occurs? Oocyte 2 will continue meiosis 2. Didn't we say that it is blocked at metaphase 2? And we know that the stages of meiosis are prophase, metaphase, anaphase, telophase. Oocyte 2 was blocked at metaphase 2, so it will finish anaphase 2 and telophase 2 to give an ootid only if fertilization happened. Okay, this ootid has N chromosomes with 1 chromatid, and then this ootid will differentiate into an ovum. This happens after fertilization. So at the beginning, when the female is still a fetus inside her mother's body, an oogonium undergoes mitosis and growth to give an oocyte 1. This oocyte 1 is blocked at prophase 1. The female is born with oocyte 1 blocked at prophase 1. At puberty, oocyte 1 does meiosis 1 and starts with meiosis 2. If there was no and stops at meiosis 2, and oocyte 2 is released into the fallopian tube. If there is fertilization, this oocyte 2 will continue meiosis 2 to give an ootid, and then differentiation of the ootid, development of the ootid will give an ovum. This ovum is now ready to be fertilized by a sperm cell. The primary oocyte is blocked at prophase 1. Secondary oocyte is blocked at metaphase 2. Now, look at the polar bodies. By meiosis 1, we were supposed to have, it's a division, right? So oocyte 1 had to divide into 2 daughter cells. But during oogenesis, only one cell is suitable to continue the divisions. The other one, which will degenerate later on, is called a polar body. It will be degenerated by the body. So we only get one secondary oocyte. Same thing. Secondary oocyte undergoes or finishes meiosis 2. We will get one polar body that will be lost and only one ootid. So it's different than the case of spermatogenesis. نحن بالspermatogenesis, we said that at the end, يعني one spermatogonium gives me 4 sperm cells. But look here, one oogonium gives me 1 ovum, and the other 3 cells are lost or degenerated because they are polar bodies. Okay? So I will repeat, this oogenesis happens in 3 phases. We have the fetal phase when the female is still in her mom's body, and all females are born with oocyte 1 blocked at prophase 1. Then at puberty, an oocyte 1 continues its meiosis, and then we get oocyte 2 blocked at metaphase 2. Only and only if fertilization happens. So after fertilization, this oocyte 2 will continue its mitosis and will differentiate into an ovum. 3 different phases. It's not a continuous process. 3 different phases. Okay? In the next video, we will be talking about folliculogenesis. Folliculogenesis and oogenesis are female processes.